Welcome to the Generative A's podcast, brought to you by BiteGoose AI and algorithmic drug discovery pipeline. Today, we are doing something I think is incredibly important, a really deep dive.
Yeah, this is a big one.
Our mission isn't just to talk about AI and drug discovery. We want to actually analyze the technical blueprint.
The nuts and bolts.
Exactly. The strategies, the specific algorithms, the data infrastructure you need to genuinely transform pharmaceutical R&D. We're trying to get hold of the engineering manual for this modern drug discovery engine.
And it's a transformation that's, frankly, overdue. It's an economic necessity now.
Because the old model just isn't working.
It's fundamentally unsustainable. I mean, look at the numbers. Costs often shooting past $2 billion for one successful drug. Timeline stretching over a decade. And the failure rate, it's still around 90% attrition across the whole pipeline.
90%.
Yeah. It tells you the process is still, characterized by slow, really expensive serendipity, just hoping you stumble onto something.
Yeah.
So the shift we're talking about today, it's towards what we call algorithmic treatment. It's a radical restructuring. Every single phase, from picking that first target molecule all the way to optimizing the final drug candidate. It's all driven by computation, by big data, by cloud infrastructure.
And the core idea is?
De-risking the pipeline right from day one. Making smarter bets earlier.
Okay. And the value for you, listening in, is that we're going to give you insight into how the cutting-edge companies are actually doing this.
Yeah, looking under the hood.
We'll deconstruct the blueprints used by leading platforms. Yeah. We'll name names Valo Health, Insilico Medicine, companies like that. We'll show you exactly how they structure those key computational phases. Target ID, hit discovery, lead optimization.
Using machine learning models that are specifically designed, really tailored, to tackle these bottlenecks that have plagued pharma for decades.
So let's kick off with that transition. This move to in silico discovery. You mentioned the 90% failure rate. That number really seems to be the stick forcing this change, right? Pushing companies away from just screening millions of compounds, hoping for a hit.
Absolutely. That failure rate is the primary driver. And AI, or machine learning more broadly, it addresses two massive, really deep-seated problems at the same time.
Okay, what's the first one?
The first is the data challenge. Historically, R&D has operated in these rigid data silos.
Right. Genomics over here, proteomics over there.
Exactly. Genomics data didn't talk to transcriptomics, which it didn't talk to proteomics, and none of them really connected properly with clinical trial data or, electronic health records, EHRs.
They were all separate worlds.
Pretty much.
Yeah.
AI provides the, let's call it mathematical scaffolding to finally break down those walls. It allows us to integrate these incredibly diverse sources, we call it multi-omics data, into unified predictive models.
And that integration is key.
It's transformative. If your model can learn from genomics and transcriptomics and real-world patient evidence simultaneously, its power to predict what actually matters for human disease goes up exponentially.
So it's not just pooling the data, it's finding the predictive mathematical links between those previously siloed data sets. Okay, what's the second challenge AI tackles?
That's the operational challenge. It's about scale and, crucially, the feedback loop. And this brings us to, I think, the defining concept of the modern tech bio company. It's this lab-in-the-loop approach.
Lab-in-the-loop, explain that a bit more.
This is where the real competitive edge lies now. You have these high-throughput, heavily automated robotic wet labs generating enormous amounts of proprietary data. But here's the kicker. The experiments they run aren't random. They are entirely designed, guided, and optimized by machine learning predictions.
Ah, so the AI tells the robots what to do.
In essence, yes, it's a closed, virtuous cycle. The algorithms say, based on our current understanding, run this specific high-value experiment. The robotics execute it perfectly, generating high-quality data.
And that new data feeds back.
Right back into the model. It makes the algorithm smarter, leading to better predictions for the next round of experiments. It means less waste, fewer dead-end experiments. The advantage isn't just the sheer volume of data, it's the AI-guided relevance and the quality of the data being generated.
Okay, so this blend of massive, unique data generation guided by computation. You're saying this is pushing the whole industry towards two big frontiers. You mentioned proximity medicine and powered genetics. Let's unpack those. What do they actually mean for future treatments?
Right. Proximity medicine. This is about designing new types of drugs, new modalities that work much more closely with the body's own existing cellular machinery. It's not just about blocking a receptor anymore.
Like what? Give us an example.
The best example right now is probably designing proteolysis targeting chimeras or PROTACs.
Or tags. Okay.
These are really complex sort of two-headed molecules. They're designed to grab onto a disease-causing protein with one end, and with the other end, they grab onto the cell's own garbage disposal system, the proteasome.
So they hijack the cell's machinery.
Exactly. To actively eliminate the bad protein, just get rid of it entirely instead of just inhibiting it temporarily. And AI is absolutely crucial here because designing a PROTAC involves optimizing so many things at once.
Like what?
You need it to bind tightly to your target protein and bind tightly to the E3 ligase part of the disposal system. And the linker connecting the two parts needs to be the right length and flexibility, and you need to ensure it actually leads to degradation. Traditional chemistry methods really struggle with that kind of complex multi-parameter optimization.
I can imagine. Okay, so that's proximity medicine. What about powered genetics?
Powered genetics is all about mastering human genetic data computationally. We have strong evidence now that if a potential drug target is genetically validated,
Meaning
that data from large human populations shows that naturally occurring variations in that gene actually reduce the risk of a specific disease, well, then a drug targeting that gene has a significantly higher chance of success in clinical trials.
So genetics points the way.
It provides incredibly strong evidence. Powered genetics is using AI to decipher these really complex, often nonlinear connections between genetic variations and disease outcomes. We use huge data sets like the UK Biobank to find these genetically validated targets.
And it's not just about finding targets.
No, it's also about using genetics to stratify patient populations for clinical trials, finding the patients most likely to respond. It's about fundamentally improving the odds of success by grounding your very first decision, the target choice, in solid human biological evidence. This systematic use of genetics is probably the single biggest factor in de-risking the early pipeline. Hashtag 1.28 taxonomy of the AI drug discovery landscape.
Okay, that sets the stage for the transformation. Now, if you look at the actual companies building these AI systems, it seems like a pretty diverse landscape. You mentioned it's segmented.
Yeah, highly segmented. We can broadly categorize companies into about 4 main strategic archetypes. And the archetype a company fits into really dictates its technology stack, its business model, how it competes.
All right, let's break them down. Category 1.
Category 1 is what I'd call the AML powerhouses. These are the companies aiming for full vertical integration. They want to reinvent the entire R&D process end to end, all in-house, from identifying the target right through to delivering a clinical candidate.
Okay, who falls into this bucket?
Yeah.
You mentioned Schrödinger earlier. What's their specific angle?
Schrödinger's deep expertise is in physics-based simulation. They build these incredibly sophisticated models combining quantum mechanics and molecular dynamics with machine learning. Their whole competitive advantage is built on predicting molecular properties, how well a drug binds, its solubility, potential toxicity with really high accuracy before anyone makes it in the lab.
So they focus on the physics simulation layer.
Very much so. Owning that predictive physics layer. Now, contrast that with a company like Exscientia.
Right, they're known more for automation, aren't they?
Exactly. Exscientia focuses on automating the entire iterative drug design cycle. That's often called the Design, Make, Test, Analyze loop, or DMTA. Their platform uses closed-loop AI optimization to dramatically speed this up. They've shown they can slash the time it takes to find an optimized lead candidate from, what used to take several years down to potentially just a few months.
That speed is obviously a huge advantage.
Massive commercial differentiator, lets them cycle through many more ideas much faster. And then you have Recurrent pharmaceuticals. They really embody that lab-in-the-loop concept we talked about.
How so?
They've built these enormous robotic screening labs combined with advanced computer vision and AI. They're generating these huge proprietary maps of biology, mostly focused on rare diseases and cancer, by running millions of experiments and analyzing the cell images. Their moat, their advantage, is less about a single algorithm and more about the sheer scale and uniqueness of the data they generate in-house.
Okay, that's the powerhouses. Category 2. You mentioned next-generation biologics design.
Yeah.
So this is about bigger molecules.
Yeah, exactly. Moving beyond traditional small molecules to large, complex biologics like proteins, peptides, antibodies. Generate. Biomedicines is a key player here.
What's their approach?
They're using generative AI, specifically large language models, or LLMs, but trained on massive data sets of protein sequences, not text. They use these models to design completely novel functional proteins from scratch proteins intended to be therapeutics.
Like generating a sentence, but with amino acids.
That's a great analogy. They're trying to solve the inverse folding problem, basically. Tell the AI the desired 3D shape and function, and have it generate the amino acid sequence that will fold into that structure and do the job. Then you have companies like Epsilon.
What do they do?
They use AI to mine the natural immune system, but at incredible scale. They can sift through millions and millions of individual B cells from humans or animals to very rapidly discover antibodies that bind tightly to a specific disease target.
Interesting. Okay. Category 3. Innovators in new modalities and platforms. These are companies built around specific biological insights.
That's right. They often have a unique technological or biological platform underpinning their work. A prime example is Intellia Therapeutics. They're focused on CRISPR gene editing.
Gene editing therapies.
Yes. And a huge part of making CRISPR work safely and effectively is designing the guide RNA, the molecule that tells the CRISPR enzyme where to cut the DNA. Intellia uses complex computation to design guide RNAs that are highly effective, but also have minimal risk of cutting in the wrong place, those off-target effects. That computational precision is vital for their therapies.
Any others in this category?
Deep genomics is another good example. They focus specifically on RNA biology, which is incredibly complex. They use AI to predict how genetic mutations affect RNA processing, like splicing, and then they design novel RNA-based drugs like antisense oligonucleotides to correct those problems. Their niche is mastering the complexities of RNA.
All right. And the final group, category 4, specialized AI and software platforms. So these are more like tool providers.
Yeah, often they excel in a specific computational area and work through partnerships or offer their platform as a service, SaaS, rather than trying to build the entire drug pipeline themselves. They're often the brains behind the scenes for bigger pharma companies.
Like who?
Companies like Ictos. They focus purely on generative AI for chemistry. They provide software tools that can rapidly design novel, but importantly, synthesizable molecules optimized against multiple criteria. They really help accelerate that light optimization phase for their partners.
Synthesizability is key there.
Absolutely. And then you have Aachen, which has a really interesting model based on federated learning.
Federated learning, how does that work in this context?
It allows them to train AI models on sensitive patient data that's spread across multiple hospitals or research centers without that sensitive data ever having to leave the original institution's servers.
Ah, so it protects privacy.
Exactly. The model travels to the data, learns locally, and only the aggregated insights or model updates are shared, not the raw patient data. They use this for things like discovering new biomarkers or optimizing clinical trial design using real-world data from diverse sources. It's a critical approach for unlocking insights from sensitive health data.
Okay, that taxonomy really helps map out the players. Now, let's dig into how these specialized tools and approaches actually get stitched together into a functioning end-to-end pipeline. You suggested we look at Valo Health and Insilico Medicine as concrete examples.
Yeah, I think they provide really good blueprints for how you actually build an AI-first R&D engine. They show how to integrate these different computational phases.
Let's start with Valo Health. Their platform is called Opal.
That's right, Opal. And it's explicitly structured to mirror the entire R&D lifecycle, but with computation woven into every single step. They emphasize what they call a human-centric data philosophy.
Human-centric,
Meaning, their architecture systematically forces the integration of multi-omics, data genomics, transcriptomics, etc., with clinical data and real-world evidence, or RWE. The goal is to ensure that whatever they're working on has demonstrable relevance to human disease right from the earliest stages.
So Opal isn't just, say, 3 separate software tools. It's designed as an integrated workflow.
Exactly. It starts with a phase they call biological discovery. This covers the initial target identification and also something they call diagnosis discovery, leaning heavily on that RWE to validate targets and understand patient populations.
Okay, after biological discovery.
Then comes therapeutic design. This is where the computational chemistry happens, everything from hit identification, finding those initial molecules, through to lead optimization, refining the best candidates.
Then the final piece
is clinical development. Here, they deploy AI for things like optimizing the design of clinical trials, stratifying patients to find those most likely to benefit, and even predicting trial outcomes. Their whole mantra seems to be fail fast, succeed smarter.
by front-loading that de-risking using computation.
Precisely. Avoid those incredibly expensive late-stage clinical failures by making sure the foundations are solid.
Okay, that's Vala's Opal. What about Insilico Medicine? Their platform is pharma.ai.
Yes, pharma.ai. And it's another excellent example, really showcasing a deep implementation of generative AI across the entire pipeline. It's built around three core interconnected engines.
Engine number one.
Is Biology 42. This is their target discovery engine. It uses a powerful mix of deep learning and natural language processing, NLP.
NLP to read scientific papers.
Exactly. It analyzes vast amounts of multiomics, data genomics, transcriptomics, proteomics, but in conjunction with information extracted from millions of scientific papers, patents, clinical trial reports. This comprehensive analysis allows it to generate novel hypotheses about disease mechanisms, build complex pathway models, and ultimately nominate high-confidence drug targets.
Okay, so Biology 42 finds the target. Then what?
Then the baton passes to the second engine, Chemistry 42. This is really the heart of their de novo small molecule design capability.
De novo meaning from scratch.
Right. Inventing completely new molecules. Chemistry 42 uses generative models, things like variational autoencoders, VAEs, or generative adversarial networks, GANs, but guided by reinforcement learning. This allows it to rapidly create novel chemical structures.
And it's optimizing for more than just binding, right?
Absolutely critical point. The optimization isn't just for potency against the target protein. The model is trained to generate molecules that are simultaneously optimized for potency and selectivity, so they don't hit other proteins, and good drug-like properties, and importantly, synthetic accessibility, meaning they can actually be made in the lab without too much trouble. The goal is to generate a high-quality lead candidate incredibly quickly.
Makes sense. And the third engine, Medicine 42.
Medicine 42 acts as that crucial final check before heading into expensive clinical trials. It's their clinical trial prediction engine.
How did that work?
It analyzes enormous amounts of historical clinical trial data, patient characteristics, and preclinical data for their candidate drug to predict the probability of success for a planned phase 2 or phase 3 trial. It's another layer of de-risking, aimed squarely at preventing those late-stage failures that can bankrupt companies.
This brings us back to something you touched on earlier, the competitive moat. If some of these foundational algorithms, maybe the basic generative models, are becoming more available, perhaps even open source, what really creates that defensible barrier for a company? What stops everyone from doing this?
That's a fantastic question, and the answer, I believe, is undeniably the data generation engine.
The wet lab part.
The proprietary high-throughput AI-guided wet lab part. You can, download TensorFlow or PyTorch tomorrow. You can access public datasets like ChEMBL, but you cannot download the unique high-quality data being generated day in, day out by a company like Recursions or Absci's highly optimized, automated robotic labs running experiments that were specifically designed by their internal AI models to fill critical knowledge gaps.
So the data itself is the moat.
It's the deepest part of the moat. Companies that lead invest heavily, not just in algorithms, but in the robotics, the novel assay technologies, the sheer infrastructure needed to generate unique data at a scale and relevance that simply doesn't exist in the public domain. Superior models aren't just about better math. They are trained on superior, unique, continuously flowing data streams generated in-house. Any winning strategy has to pair the AI development with a parallel proprietary data generation strategy. That's the long-term competitive lock-in.
Okay, let's zoom right into that critical first phase then. Tidy Valo, target identification and validation. You said the mission here isn't just finding a protein that looks interesting.
No, absolutely not. You have to answer two really specific hard questions. First, is this protein likely to be therapeutically actionable? Can we actually drug it effectively? And second, maybe even more important, how strong is the causal, human-relevant evidence linking this target to the disease?
Because if you get this wrong,
the whole project sinks. All the downstream investment in chemistry, in preclinical studies, and clinical trials, potentially hundreds of millions, even billions of dollars, it's all wasted if you started with the wrong target. So modern AI approaches aim to build a really strong case for validation through triangulation.
Triangulation, meaning evidence from different angles.
Exactly. Pulling evidence from multiple, independent, orthogonal sources to build confidence. And that triangulated evidence usually starts with human genetics. Genomics-driven target discovery often provides the highest level of initial validation. Hashtag tagged 3.2 genomics-driven target discovery.
Why is genetics so powerful here?
Because it directly connects variations in a gene to the risk of developing a disease in actual human populations. We rely heavily on massive data sets, like the UK Biobank with its 500,000 participants. We use these for genome-wide association studies, GWS.
Right, GWSS. Those studies link broad regions of chromosomes to disease risk. But there's a challenge there, isn't there? They don't pinpoint the exact cause.
That's the bottleneck. A GWS might flag a large region, maybe hundreds of thousands of DNA base pairs long, that contains potentially thousands of tiny variations, these single nucleotide variations, or SNVs. We need to figure out which single SNV within that large region is the actual culprit driving the disease risk.
How do you do that?
Well, often the causal SNV isn't even inside the protein coding part of a gene. It's in the non-coding DNA, the regulatory regions. These regions act like switches bound by proteins called transcription factors, or TFs, that control how much a gene is turned on or off.
So a bad SNV messes up the switch.
Precisely. If the SNV disrupts the binding of a key TF, it can dysregulate the gene's expression level, leading to disease. To find these critical SNVs and quantify their impact, we use a specialized type of deep learning model, the 1D convolutional neural network, or 1D CNN.
Okay, CNNs are usually for images, right? How does a 1D CNN work on a DNA sequence, which is like text?
You treat the DNA sequence, the A's, T's, C's, and G's, as a 1D input signal, often one-hot encoded. Then you train the 1D CNN using experimental data, like ChEMBL data, which tells you exactly where specific TFs actually bind across the entire genome in real cells.
So the CNN learns from real binding events.
Yes. The filters, or kernels, of the 1D CNN slide along the DNA sequence, and they learn to recognize the specific short patterns, the sequence motifs that are characteristic of TF binding sites. It essentially learns the grammar of gene regulation for that specific TF. Once trained, the model gets really good at predicting the probability that any given short stretch of DNA is a functional binding site.
Okay, so how do you use that trained model to find the causal SNV from that big GWA's region?
You run a comparison. For each candidate SNV in the region, you take the DNA sequence with the common version of the DNA letter, that's the wild-type allele, and you feed it to the model to get a predicted TF binding probability. Then you swap in the sequence with the alternative letter, the mutant allele containing the SNV, and you run it through the model again to get a new binding probability.
And you compare the two probabilities.
Exactly. The difference between those two scores, sometimes called the variance score, tells you how much that single letter change is predicted to impact TF binding. If the SNV causes a big drop, or sometimes a big increase, in predicted binding, that variant gets flagged as potentially causal.
And the gene regulated by that disrupted binding site becomes your target.
It becomes a very high-confidence, genetically validated potential drug target. You've moved from just a statistical association in a GWAS to a specific, testable hypothesis about the molecular mechanism involving TF binding disruption, #tag3.3 transcriptomics and multi-omics via denoising autoencoders.
Okay, so genomics gives us that strong causal link, the why.
Yeah.
But we also need to understand what's actually going wrong inside the diseased cells functionally. That seems like the job of transcriptomics, looking at gene expression.
That's right. Transcriptomics tells us which genes are turned up or down in disease versus healthy states. But as you hinted, this data is notoriously challenging to work with.
Why is that?
Two main reasons. First, it's incredibly high-dimensional. You're typically measuring the expression levels around 20,000 genes. And second, it's really noisy. You get technical noise from the sequencing process itself, and you get biological noise because gene expression naturally fluctuates quite a bit even in identical cells.
So lots of genes, lots of noise. How do you cut through that?
This is where denoising autoencoders, or DAEs, become incredibly useful. A DAE is a type of unsupervised neural network that's specifically designed to learn robust representations from noisy data.
How does it denoise?
The trick is during training. You take a clean gene expression profile, you intentionally corrupt it maybe by adding random noise or masking out some of the gene values, and then you train the network to reconstruct the original clean profile from that noisy input.
Ah, so it has to learn to ignore the artificial noise.
Exactly. And to do that effectively, the network architecture forces the data through a bottleneck, a hidden layer, with far fewer nodes than the input or output layers. This is the latent representation.
The compressed version.
Right. To successfully reconstruct the full, clean 20,000 gene profile, the encoder part of the network has to learn to discard the random noise and capture only the essential underlying biological signal in that compact latent space.
And what do those nodes in the latent space represent? not individual genes, presumably.
No, that's the really powerful part. Those latent variables often end up corresponding to coordinated biological programs or pathways that govern the cell state, things like the MAPK signaling pathway or the glycolysis metabolic pathway, or maybe a specific immune response signature.
So you get pathway-level insights.
Precisely. By comparing where disease samples map in this latent space versus healthy samples, you can very clearly see which fundamental biological pathways are dysregulated in the disease. This immediately highlights high-priority targets within those pathways. And it's a great way to integrate multiomics data. You can train DAEs on transcriptomics, proteomics, metabolomics, and find these core underlying biological themes. It's key for biomarker discovery, too. Hashtag 3.4 target deconvolution via perturbation barcodes.
Okay, so now we have a target that's potentially genetically validated, and we see the pathway it's involved in is dysregulated based on transcriptomics or multiomics. What's the final piece of this TDVL triangulation?
The final piece is the functional check. Can we show that modulating this target or pathway actually reverses the disease state, at least in a relevant cell model? This is where we leverage massive public datasets from perturbation screens, especially the LINCS L1000 pound dataset.
LINCS L1000 what is that exactly?
The Library of Integrated Network-Based Cellular Signatures, LINCS L1000 pounds project, measured how the expression levels of about 1,000 carefully chosen landmark genes change in response to thousands of different perturbations, adding drugs, knocking down genes, etc. The clever part is that from those 1,000 landmark genes, you can computationally infer the expression changes for the rest of the 20,000 genes in the transcriptome.
So it's an efficient way to get a snapshot of the cell's response.
A very efficient way to profile millions of perturbations. Now, to make sense of all these profiles, we use another specialized deep learning architecture, often a Siamese network.
Siamese network, like twins.
Kind of. You train it to take any L1000 pounds expression profile, whether it represents the effect of a drug or maybe a gene knockdown or even a disease state, and condense it down into a fixed-length numerical vector, a barcode.
A unique fingerprint for that cellular state. Why use a Siamese network for that?
The Siamese architecture is trained specifically to ensure that similar biological states produce similar barcodes, and dissimilar states produce barcodes that are far apart in the vector space. It learns a meaningful distance metric. This makes the barcodes reliable and comparable representations of the underlying biology.
Okay, so you have barcodes for thousands of drugs, and maybe a barcode for your disease state. How do you use them?
Here's the crucial step for validation and even drug repurposing. You take the barcode representing your disease signature, that pattern of dysregulated gene expression. Then you search through the huge library of L1000 pounds barcodes generated from known drugs, looking for compounds whose barcodes are strongly anti-correlated with the disease barcode.
Anti-correlated, so the exact opposite pattern.
Exactly. You're looking for a drug that induces the mirror image transcriptional state compared to the disease. Finding such a drug provides strong functional evidence that modulating the pathways represented by that signature can indeed reverse the disease phenotype, at least at the cellular level.
And this confirms the target's relevance.
It provides that crucial third leg of the stool. It's powerful for confirming your primary target's mechanism, and it's fantastic for finding existing drugs that could potentially be repurposed for your disease.
So that triangulation, the genomics for the causal link, the DAEs on omics data confirming pathway dysregulation in patients, and the L1000 pound barcode showing functional reversal in cells that constitutes a really well-validated, de-risked target to take forward.
That's the gold standard for starting an AI-driven drug discovery project today.
All right, we have our high-confidence validated target. Now we need to actually find a molecule, a drug, that interacts with it. This is the hit discovery phase.
Yeah.
You mentioned the chemical space is unbelievably vast.
Astronomical is the word. The estimated number of possible drug-like small molecules is around 1060, 10 followed by 60 zeros. It's bigger than the number of atoms in the universe. Trying to explore that space randomly is just futile.
So computation is the only way. How do platforms approach this huge search problem systematically?
They typically employ a combination of 3 core computational modalities, leveraging the strengths of each. Think of it as a triad: virtual screening, de novo design, and physics-based simulation.
Okay, let's take those one by one. Virtual screening. This sounds like the most established method.
It is. Virtual screening is essentially searching through libraries of known chemical compounds. You might take a database like ZINC, which contains billions of commercially available molecules, and computationally dock each one into the 3D structure of your target protein to predict if it might bind.
What are the pros and cons?
The big pro is speed and synthetic accessibility. You can screen literally billions of compounds computationally in maybe a day or so, and if you find a hit, you can just buy it off the shelf and test it in the lab immediately.
But the downside?
The major con is coverage. Even billions of molecules is a minuscule fraction of that 10 total chemical space. You're limited to chemical structures that someone has already made or thought of. You might completely miss out on truly novel, effective chemical scaffolds.
Right. Which brings us to de novo drug design. This is about inventing completely new molecules.
Exactly. De novo means from new. Instead of screening existing molecules, you use algorithms to generate or invent entirely novel chemical structures that are predicted to have the properties you want. It tackles the inverse problem. Tell me the molecule that has property X rather than tell me the property of molecule Y.
This is where generative AI really comes in.
Absolutely. Generative models are incredibly powerful for exploring that vast, uncharted chemical space efficiently. They can construct novel molecules atom by atom or fragment by fragment. This is crucial if you're looking for a first-in-class drug with a completely new mechanism, and it helps build strong intellectual property.
Sounds great. Is there a catch?
The main challenge historically, and still to some extent, is synthetic feasibility. Generative models, left completely unconstrained, can sometimes dream up molecules that look fantastic on the computer but are incredibly difficult or even impossible to actually synthesize in a chemistry lab.
So the generated molecule might be impractical.
It could take years and dozens of complex steps. So modern de novo design platforms have to incorporate prediction of synthetic accessibility right into the generation process. They optimize not just for binding, but also for makeability.
Okay, so VS for known space, de novo for unknown space. What's the role of the third modality, physics-based simulation?
Physics-based methods like molecular dynamic simulations or solving the Schrödinger equation for quantum mechanical effects act as the ultimate reality check.
High fidelity, but slow.
Extremely high fidelity, but computationally very expensive. We're talking potentially days or even weeks of supercomputer time for a single accurate calculation on one molecule complex. So it's completely impractical for screening millions or billions of compounds.
So where do you use it?
You use it strategically, usually later in the process, on a smaller number of promising candidates identified by VS or de novo methods. You use it to refine the exact 3D pose of how the drug sits in the protein's binding site, to calculate a really accurate binding free energy, that's the delta G, to reliably rank your best candidates, or maybe to understand exactly how a small change to the molecule impacts its binding.
So the optimal pipeline uses all three.
Definitely. It's usually a flexible hybrid approach. You use AI-driven methods like VS and de novo design for speed, for exploring space, for generating hypotheses, and then you use the rigorous physics-based simulations for high-fidelity validation and refinement of your top contenders. Hashtag 4.1 representing molecules for AI.
Before we dive deeper into the generative algorithms themselves, we need to talk about how AI actually sees a molecule. It needs numbers, not chemical drawings. How do you represent molecules numerically?
That's a fundamental question, and the representations we use have evolved quite a bit. One common, older way is the SMILES string.
Smiles.
Simplified Molecular Input Line Entry System. It's a text-based way to represent a molecule's structure linearly. For example, aspirin is CC, O1C, CC, C1C. It's like a chemical sentence.
And AI models can process text strings.
Right. Sequential models, like recurrent neural networks, RNNs, were often trained on SMILES strings because they treat the molecule like a sequence of characters. However, a more powerful and arguably more natural representation, especially for modern AI, is the molecular graph.
A graph, like nodes and edges.
Exactly. You represent each atom as a node in the graph, and the chemical bonds between atoms as the edges connecting those nodes. The nodes and edges can have features describing the atom type, carbon, nitrogen, etc., or bond type, single, double.
Why is the graph representation considered better?
Because it directly captures the molecule's topology, how the atoms are connected, which is fundamental to its chemical properties. It's the native language for graph neural networks, GNNs, which have become incredibly powerful in chemistry.
How do GNNs work on these graphs?
GNNs work by passing messages between connected nodes, atoms, in the graph. Each atom learns about its local chemical environment by receiving information from its neighbors. By repeating this message passing, the GNN can learn very complex features related to the molecule's structure, like ring systems, functional groups, and even 3D spatial relationships, often more effectively than models working on the linear SMILES string, which can struggle with long-range dependencies in the structure.
Okay, so we have our molecular representations. Now let's look at the generative models used for hit discovery, for inventing those new molecules. You mentioned RNNs working on smiles. How do they generate new chemistry?
You train an RNN, often a type called an LSTM or GRU, on a massive database of known drug-like molecules represented as smile strings, maybe from ChEMBL. The RNN learns the statistical patterns, the grammar of valid chemical structures. It learns, for example, that a carbon atom typically forms 4 bonds, oxygen forms 2, and so on.
It learns the rules of chemistry from examples.
Exactly. Once trained, you can sample from the model, you give it a starting character, and it predicts the next most likely character, then the next, and so on, generating a novel smile string, character by character, that hopefully corresponds to a valid new molecule. The initial model is like a general chemist.
But you mentioned something called transfer learning makes this much more powerful for specific projects.
Yes, this is a key technique. You take your general RNN model, which has learned the broad rules of chemistry from millions of examples, and you fine-tune it. You continue training it, but now on a much smaller data set containing only molecules known to be active against your specific target protein.
So you specialize the generalist model.
Precisely. Even if you only have, say, 20 or 50 known active compounds for your target, fine-tuning the pre-trained RNN on this small set strongly biases its generation process. It starts generating new molecules that are not just generally drug-like, but specifically resemble the structures known to bind your target.
The result being?
The result is what's called a focused library. You generate a set of novel chemical ideas that are much more likely to be active against your specific target compared to just randomly sampling from the general model. It dramatically increases the hit rate of your generated molecules.
Okay, that's the RNS Miles approach with transfer learning. What about the other main generative architecture you mentioned? Adversarial autoencoders, or AAEs. How do they work differently? They use that latent space idea.
Right. AAEs, and also similar models like variational autoencoders, VAEs, work by learning to map molecules into and out of a continuous, usually low-dimensional mathematical space called the latent space. The encoder network compresses the input molecule, often represented as a graph or fingerprint, into a point in this latent space. The decoder network learns to take a point from the latent space and reconstruct the original molecule.
And the adversarial part in AAE, what does that do?
The adversarial component is a discriminator network. Its job is to look at the points being mapped into the latent space by the encoder and try to distinguish them from points sampled from a simple prior distribution, like a standard Gaussian, a bell curve. The encoder is trained to fool the discriminator.
Why try to fool it? What does that achieve?
It forces the encoder to structure the latent space smoothly and continuously, making it resemble that simple prior distribution. This smooth structure is incredibly valuable. It means that points close together in the latent space correspond to molecules that are structurally similar.
And why is that smoothness so useful for drug design?
Because it enables optimization. If the
Yeah.
space is smooth, you can define a property you want to maximize, maybe predicted binding affinity or solubility or novelty, and you can mathematically walk through the latent space in the direction that increases that property. You perform gradient ascent in the latent space.
So you can navigate the space to find better molecules?
Exactly. You find a new point in the latent space optimized for your desired properties, and then you feed that point to the decoder network, which generates the corresponding novel molecule. It turns molecule generation from a somewhat random search into a more directed optimization process, giving you much more control.
Okay, we've used generative models, maybe combined with virtual screening, to find some initial hits molecules that bind our target. Now we need to turn those hits into actual drug candidates.
This is lead optimization, right?
Yes, phase 3, lead optimization. And the goal here shifts significantly. We're no longer just looking for any molecule that binds. We need to take a promising hit structure and meticulously refine it. This is all about multi-parameter optimization.
Multiple parameters. Like
We need to maximize potency, make it bind really tightly. We need to improve selectivity, make sure it binds only to our target and not other related proteins, which could cause side effects. And critically, we need to optimize its ADMET properties.
absorption, distribution, metabolism, excretion, and toxicity. Basically, does the molecule get into the body, go where it needs to go, not get broken down too quickly, get cleared properly, and is it safe? Optimizing all of these things simultaneously is the core challenge of lead optimization.
And computation plays a big role here too, I assume. What kinds of models are used?
The models need to become more quantitative and structure aware at this stage. Two important examples are 3D convolutional neural networks, 3D CNNs, and atomic convolutional networks, ACNs.
Let's start with 3D CNNs. We talked about 1D CNNs for DNA. How do 3D CNNs work for molecules?
They work on the 3D structure of the drug molecule bound within the protein's active site. You take this 3D complex and represent it as a grid of voxels, like pixels but in 3D.
So you turn the protein drug complex into a 3D image.
Essentially, yes, a multi-channel 3D image. Each channel in this voxel grid represents a different physicochemical property at that point in space. For instance, one channel might map out where the hydrogen bond donors are, another channel maps hydrophobic atoms, another maps positive or negative charges, maybe metal ions.
Okay, and the 3D CNN processes this grid.
Right. The 3D convolutional filters slide through this 3D volume, and they learn to recognize specific 3D patterns that are important for binding. For example, a filter might learn to recognize the perfect geometric arrangement for a strong hydrogen bond between the drug and a specific amino acid in the protein pocket.
So it learns favorable 3D interactions. How is this used in optimization?
It allows for very rapid prediction of how small changes to the drug molecule might affect its binding pose and its interaction score. You can computationally explore modifications to your lead compound and quickly prioritize changes that the 3D CNN predicts will improve shape complementarity or add favorable interactions within the binding site.
Okay, that's 3D CNNs for structural patterns. What about atomic convolutional networks, ACNs? You said they aim for predicting the actual binding energy.
Yes, ACNs are designed to predict the binding free energy, the delta G doi, which is the fundamental thermodynamic measure of binding affinity. Higher affinity, more negative delta G doi, generally means higher potency.
How do they achieve that level of quantitative prediction?
A key innovation in many ACN architectures is how they incorporate the thermodynamic cycle directly into the learning process, instead of just training the model to predict the final delta G value directly from the complex structure.
What do they do instead?
They train the model to predict the energy of the bound complex and the energy of the isolated protein and the energy of the isolated ligand drug molecule, all separately but using shared network components. The model's prediction for delta G day is then calculated from the differences between these predicted component energies. Delta G complex, G protein, D ligand. The model is trained to make this calculated delta G DA match the experimentally measured value.
Why force it to learn the separate energies? That sounds more complicated.
It forces the model to learn more physically meaningful representations. By having to predict the energies of the individual components as well as the complex, the ACN is implicitly pushed to learn the underlying non-covalent interactions, hydrogen bonds, van der Waals forces, electrostatics that actually determine binding affinity rather than just memorizing surface correlations.
So it learns the physics better.
It learns representations that are closer to the underlying physics. ACNs are also often designed to be intrinsically rotationally and translationally invariant, meaning the prediction depends only on the relative positions of atoms, not the overall orientation of the molecule in space, which is physically correct. This makes them more robust and generalizable. They really represent a powerful bridge between physics-based principles and deep learning for quantitative prediction. #tag 5.3 real-world case studies.
This all sounds incredibly powerful in theory, but let's talk results. Can you give some real-world examples where these AI approaches have genuinely made a difference? Maybe starting with that antibiotic, Halcin.
Halcin is a fantastic example. It came about directly because of the growing crisis of antibiotic resistance. Researchers at MIT wanted to find completely new types of antibiotics.
How do they use AI?
They used a type of graph neural network called a directed message passing neural network, or DMPNN. They trained this GNN on a relatively small but high-quality data set of molecules that were already known to inhibit the growth of the bacterium E. coli.
So they taught the model what an E. coli inhibitor looks like chemically.
Exactly. Then they took this trained model and used it to screen a huge virtual library over 107 million different molecules from the ZINC database.
And the model picked out Halcin.
It picked out several promising candidates, but Halcin stood out. It was a molecule that had been investigated before for diabetes, but never as an antibiotic. And structurally, it looked completely different from any existing class of antibiotics.
And did it work?
Amazingly well. Experimental testing confirmed it was a potent, broad-spectrum antibiotic. Crucially, it was effective against many strains of bacteria that had already developed resistance to multiple existing drugs, including some really dangerous pathogens like Clostridium difficile and Acinetobacter baumannii.
Wow. So the AI found a truly novel structure with potent activity.
Yes, it demonstrated the power of these GNNs to learn complex structure-activity relationships from limited data and then generalize that knowledge to rapidly identify a first-in-class clinical candidate from an enormous chemical space.
That's impressive. What about the speed aspect? You mentioned the gingerol platform and a kinase inhibitor.
Right, the GeneRel case study, which involved Insilico medicine and others, is all about the speed of optimization. They were targeting a protein called DDR1 kinase, which is implicated in fibrosis and other diseases.
What was their AI approach?
They used a combination approach. A variational autoencoder, VAE, was used to learn that smooth, latent space representation of relevant chemical structures. Then, they used reinforcement learning, RL. The RL agent's job was to navigate through the VAE's latent space, searching for points that corresponded to molecules maximizing a complex reward function.
And the reward wasn't just potency.
No, it was a multi-parameter reward. It balanced predicted high potency against the DDR1 target, good predicted synthetic accessibility, and also chemical novelty. They wanted structures that were different from known DDR1 inhibitors.
Okay, so the AI designed novel, potent makeable molecules. What was the timeline?
This is the truly stunning part. The entire cycle from kicking off the generative AI run to design the molecules, then actually synthesizing the top candidates in the lab, and finally getting experimental confirmation in cell-based assays that the compounds were indeed active against DDR1 took only 21 days.
21 days. From design to validated hit.
21 days. It's a landmark result. It provides concrete proof of how effectively AI, particularly this combination of generative models and reinforcement learning, can compress the traditionally months or years-long bottleneck of iterative lead optimization down to just a few weeks.
That kind of time compression must be completely changing the economics and possibilities of drug discovery.
It absolutely is. It's revolutionary.
We've talked a lot about algorithms and models, and you emphasize that proprietary data is the ultimate competitive advantage. But even companies generating their own unique data don't start from scratch, right? They must build on public data resources.
Oh, absolutely. Algorithms are completely useless without high-quality data to train on. And while proprietary data is key for differentiation, every platform needs to systematically ingest, clean, and integrate several cornerstone public data sets. They provide the essential foundation and context.
Okay, so if you were building a new AI drug discovery platform today, what are the essential public data sets you'd absolutely need?
There are several critical ones. First, as we discussed for Tidival, you need human genetics data. The UK Biobank is paramount here. Genetic and longitudinal health data on 500,000 people. That's the bedrock for training those 1D CNNs for variant effect prediction and finding those genetically validated targets.
Makes sense. What else?
For the chemistry side, ChEMBL is indispensable. It's a huge, high-quality, manually curated database from the European Bioinformatics Institute containing over 2 million bioactive molecules with reported activities against various targets.
What do you use ChEMBL for?
Two main things. First, it's essential for training general-purpose models that predict drug properties, like ADMET models. Second, it serves as the massive pre-training corpus for your generative chemistry models, like the RNNs or VAEs. You teach them the basic rules of drug-like chemistry using ChEMBL before you fine-tune them on target-specific data.
Okay, UK Biobank for genetics, ChEMBL for bioactivity. What about sourcing molecules for screening?
That's where the ZINC database comes in. It's a massive collection, now containing literally billions of chemical compounds that are commercially available for purchase. It's the primary source for molecules when you're doing large-scale virtual screening.
Got it. Any others, maybe for the more quantitative models?
Yes. For training models that predict precise binding affinities, like those atomic convolutional networks, ACNs we talked about, you need quantitative data. BindingDB is a key resource here. It aggregates experimentally measured binding affinity data, like KD, IC50 values, from patents and literature. It's considered a gold standard data set for training those high-fidelity delta GAG prediction models. Hashtag, tag 7.2, the importance of cloud architecture.
So you're pulling in these massive public data sets, plus generating potentially petabytes of your own proprietary omics data, image data from robotics, simulation results. Managing all this must require serious infrastructure. You can't just run this on a laptop.
Not even close. A scalable, robust cloud architecture isn't just nice to have, it's absolutely non-negotiable for running an operational AI drug discovery pipeline today. The sheer volume and heterogeneity of the data demand it.
What does that architecture typically look like? Are we talking data warehouses?
Actually, for this kind of scientific data, a data lake architecture is generally preferred over a traditional data warehouse.
Data lake versus data warehouse. What's the key difference?
A data lake, often built using cloud storage services like Amazon S3 or Google Cloud Storage, is designed to hold massive amounts of data in its raw native format. You can dump raw sequencer output files like FastTQ or BAM,
Hmm.
huge microscopy images, unstructured text from papers, alongside structured database tables, all into the same place relatively cheaply.
Is it more flexible for diverse data types?
Exactly. You don't have to force everything into a rigid predefined schema before you store it. You apply structure later when you need to query the data that's called schema on read. You use tools like AWS Glue or Google Dataproc Data Catalog to discover or define schemas and make the data queryable. This flexibility is crucial when you're dealing with constantly evolving experimental assays and data formats in R&D.
Okay, so data storage is handled by the data lake. What about the heavy computation, especially for things like processing the genomics data?
Right. Analyzing raw sequencing data, doing alignment, variant calling is incredibly compute intensive. You absolutely need automated, scalable pipelines for this. Cloud platforms offer tools perfectly suited for this.
Such as?
You typically use workflow orchestration tools like AWS Step Functions or Google Cloud Workflows to define and manage the complex multi-step analysis pipeline. Each step in the workflow, say alignment using BWA or variant calling using DTK, is usually packaged up as a software container using Docker.
And where do these containers run?
They run on managed batch computing services like AWS Batch or Google Batch, which can automatically provision and scale the necessary compute resources, CPUs, GPUs, memory, needed for each step, often using cheaper spot instances. This allows you to process hundreds or thousands of genomes in parallel, automatically and cost-effectively. The results, like the final VCF files containing the variants, are then written back into the data lake, ready for the downstream AI models like the T-Val engines to consume.
So the cloud provides the scalable storage, the workflow automation, and the on-demand compute power needed to make this whole AI pipeline function at scale.
Precisely. Without that robust cloud foundation, the algorithmic blueprints we've been discussing would remain purely theoretical. The infrastructure enables the science.
Wow. Okay, this has been a truly deep dive. We've covered so much ground, from why the shift to AI is happening, the different types of companies in the ecosystem, to detailed breakdown of the computational pipeline phases like TD-Val,
Through to the specific algorithms like 1D CNNs for genomics, DAEs for transcriptomics, the generative models like RNNs and AEs for creating molecules.
and then the optimization models like 3D CNNs and ACNs, backed up by those incredible case studies like Hallison and Gentrell, and finally the data and cloud infrastructure holding it all together.
I think the central message, the key takeaway for you listening, is that this shift to AI is fundamentally changing drug discovery from what was often a somewhat generalized trial and error process into a much more precise, rational, engineered discipline.
Engineered being the key word there.
Yeah. We're now systematically integrating deep insights from human genetics, from complex multi-omics data, using sophisticated generative chemistry tools, all to design treatments in a much more targeted and methodical way.
And the tangible impact is already clear. We see it in the success of companies like Valo, Insilico, Exscientia, Recurrent, and especially in that dramatic compression of timelines, taking discovery work that used to take years and squeezing it down to months, or, like in the Gentrell case, literally just weeks. That's revolutionary.
It truly is. And that speed, that increasing capability, brings us to a final, maybe provocative thought to leave you with.
Okay, let's hear it.
Most of the systems we've discussed today represent what we might call augmented drug design. The AI is incredibly powerful, but it's still primarily assisting human researchers, augmenting their capabilities.
Right. Humans are still in the loop.
But the clear trajectory, the direction the field is moving in, is towards autonomous drug discovery. Imagine AI systems that don't just suggest experiments, but actually make the iterative design, make, and test decisions entirely on their own, directly controlling fully automated robotic laboratories.
A closed loop with no human intervention.
Potentially. An AI that identifies a target, designs the molecule, instructs robots to synthesize it, instructs other robots to test it, analyzes the results, and then designs the next molecule, iterating completely autonomously.
That's a profound shift.
It is. And if we're heading towards that future, where AI could take the driver's seat entirely in developing our next generation of medicines, we absolutely need to start thinking now. What are the ethical considerations? What are the regulatory guardrails we need to put in place before that happens? Because that level of autonomy changes absolutely everything.